Good afternoon, everyone, and thank you for your kind introduction. And I would like to thank organizers for giving me opportunity to present our two of our products, Serum Institute, who is continuously working on elimination of TB, and we are having two good products. And I will be talking on basically global TB eradication and disease burden reduction and elimination strategy. So all of you are aware that WHO has a goal of eliminating TB by 2030. It is an optimistic goal, but definitely we can achieve this with different strategies that we can incorporate in our national programs. So I will be talking on over the next 30 minutes on TB epidemiology, the worldwide TB epidemiology, then global TB milestones and targets. What are the targets and the milestones that are set by WHO, United Nations, and where are we standing on that? Then the strategy for the TB elimination that we are coming up with: detection, treat, and prevent. Then what all, for detecting latent TB infection, what all diagnostic tests we have. Then one such test recently approved by EMA, European Medical Agencies, is CyTB. This is a new skin test. I will be talking on briefly the clinical development of the CyTB. And then there are various TB vaccines, new TB vaccines in the pipeline. Most of them are in clinical trials. Some of them are in advanced phase 3 clinical trials. One such candidate that Serum is working on is VPM1002. It is a RBCG, that is recombinant BCG vaccine. So recently we completed 2 large phase 3 clinical trials with this vaccine. So I'll be talking on the clinical development of this vaccine. And lastly, how this strategy of detect latent TB, treat latent TB, and prevent the TB disease, how that can be implemented, I will be talking. And lastly, the conclusion. So as you are aware, as per WHO TB report 2024, during 2023, so before COVID, TB was one of the largest cause of death as a single infectious agent. But during COVID, since 2020, 21, 22, for these 3 years, the COVID took that position, and it contributed to large number of deaths as an infectious disease. During this 3-year period, again, the TB epidemiology changed all over the world because of the underreporting of the TB incidence as well as TB deaths. And since 2023, there is a steep, again, rise in the reporting of TB cases as well as TB deaths. Still, there are some countries where the reporting is not come to the pre-COVID level. And there are certain countries, but since 2023, again, TB has taken that position as a leading cause of death in terms of infectious diseases. And it is estimated that during 2023, more than 10 million annual TB cases and around 1.25 million annual TB deaths reported. So these are very huge numbers. And both all the members of United Nations as well as World Health Organization, they have taken initiative and the urgent action required for end the global TB epidemic by 2030. So this is, again, we are in 2025, so we have 5 years to achieve these targets. How can we achieve? So everyone is perplexed that, is it really possible? Whether WHO is more of an optimistic, but definitely if all the countries, particularly the countries where there is a high burden of TB disease, if they implement certain strategy, then it is possible, not impossible. So this is a diagram. Basically, it shows the high TB burden countries. So these are total 49 TB countries, 49 countries. They are divided in different quadrants. So those with having high TB burden, then TB HIV-associated TB, and where the MDR, that is multi-drug resistance and rifampicin resistance TB. So basically, this quadrant shows that these are the high burden TB countries with having MDR and rifampicin resistance. This quadrant shows countries with high TB burden along with HIV-associated TB. So the Thailand falls into this quadrant, that high TB burden as well as HIV-associated TB. And this was during the period of 2021 and 25. So as per a WHO report, the end TB strategy, as per end TB strategy, what was the goal? So they had a goal as compared to 2015. In next 10 years, by 2025, we will achieve certain targets. Where are we in terms of those targets? So in terms of TB incidence rate, it was expected that there would be around 50% reduction by 2025 as compared to 2015, but we are just 8.3%. Then number of TB deaths, we were expecting that 75% reduction, but by 2023, we are achieving only 23% reduction. And in terms of percentage of TB affected households facing catastrophic costs, so there we are not seeing any improvement. So still the countries are spending money on the TB treatment and the other programs. Again, this is 2023 United Nations high-level meeting on TB and the targets for the 2027. Here, I would like to bring to your notice about the TPT coverage, that is TB preventive therapy for the household contacts. Though the target is 90% by 2027, by end of 2023, we are having only 21% TPT coverage. And WHO endorses that prevention of latent TB infection, the treatment of latent TB infection is one of the important cornerstone in the TB elimination strategy. So we need to achieve this, and for this, definitely we need diagnostic tests which are more specific, and we give the TPT to the population which desire to, require to get that treatment. Another is the new TB vaccine. So the BCG is the only vaccine that has been approved for the prevention of TB. And the BCG, it's almost more than a decade, and it was the, in fact, BCG was the first vaccine that was developed. And BCG is basically only, it's given at birth, and it prevents the severe form of TB disease, like TB meningitis in children up to 5 years of age. And the BCG has not shown any effectiveness in adults in pulmonary TB as well as extrapulmonary TB. Therefore, there is a continuous drive to develop a new TB vaccines which are more effective, and they will also prevent the adult TB. So they are saying that currently there are 6 new TB vaccines in the clinical trial, advanced clinical trial, phase 3, and expecting that in the next 4-5 years, we will be implementing some of these vaccine candidates in the immunization programs. So yes, to achieve the target of TB elimination by 2030, we have to come out of the box, okay? TB, we know the TB bacilli is a notorious bacilli. It remains in the lungs even after the completion of treatment. The bacilli remains dormant in the lungs, and we do get the relapse cases, true relapse cases, even after successful treatment. And most of them, when they have the relapse, these are the multi-drug resistance TB cases. And so we have to be think in terms of multi-pronged approach, that not only the vaccines, effective vaccine will be helpful in TB elimination, or newer antibiotics that we develop that will help in this strategy, elimination of TB. But we have to think that the latent TB infection, if you can detect latent TB infection, Then once we detect the latent TB infection, treat the latent TB infection, because latent TB, these are asymptomatic, they carry the TB bacilli, though they are asymptomatic, but during their lifetime, there is always a chance of having an active TB disease, and once they develop active TB disease, they are going to spread the disease. So we have to reduce the TB reservoir, TB bacilli reservoir. So one of the reservoir is the latent TB infection. So detect latent TB infection, treat it. So treat it with isoniazid rifampicin. And once they are treated, then give them the vaccine. So give them some effective vaccine so that they are protected, so they don't again get infected with the TB bacilli, whether it is latent TB infection or maybe later on becoming active TB disease. So this strategy of detect latent TB, treat latent TB, and prevent TB by vaccination. This can definitely break the spread cycle of the TB and reduce the reservoir of the TB bacilli in the community and help in achieving our goals. So around 25% of the world's population are latently infected with TB. And most of them are, again, similar to TB epidemiology. These are in the developing countries. And these are latently infected patients, as you are aware, they are healthy. But during lifetime, 5 to 10% of them progress to active TB disease, and then they spread the disease. Therefore, testing and treatment of latent TB infection will prevent the progression of latent TB to active TB, and they will stop the spread cycle. So coming to the latent TB diagnostic test, so we know that since long time, since many years, we are using PPD or the TST, tuberculin skin test, for the diagnosis of latent TB infection. But latent PPD has definitely certain disadvantages, and therefore there is always a drive to have a new diagnostic test for the latent TB. So one such test is IGRA, or the QFT cold or QFT test that we say. So this is a blood test where that is based on the two protein, ESAT6 and the CFP10. So we take the blood sample and it is tested in the lab. And if there are cutoff values, 0.35 milli international unit. If it is more, then it is positive. Based on that, we can consider as latent TB infection. So the PSI-TB, basically PSI-TB is a new generation skin test. It has the simplicity of PPD skin test, so it can be used in the field. So it is again similar to PPD. It is given intradermally in the forearm, and after 2 to 3 days after injection, we look for the induration at the injection site. And if it is beyond cutoff, so if it is, then it is positive. And moreover, it is more specific because it is again PSI-TB is based on these two antigens, RD, ESAT, and CFP10. So RD, ESAT6 and CFP10, this shows that's the red. This is basically, these are antigens specific to Mycobacterium tuberculosis. While PPD, it is a crude mixtures of antigens from the Mycobacterium tuberculosis as well as non-tuberculous mycobacteria, as well as the BCG, so Mycobacterium bovis BCG vaccine strain. That's why the interpretation with the PPD is always a challenge. So if someone is BCG vaccinated, then PPD can give the false positive results. And again, the individuals with immunocompromised individuals, there we have a different cutoffs. So PPD has a different cutoffs for the healthy individuals, like 10 millimeters. If they are immunocompromised, it is 15 millimeters. So again, with the CD4 count in HIV positive, with the CD4 count, that criteria changes. So clinicians always find difficult how to interpret the results for the PPD. So the PSI-TB, it is very specific. It is not affected by, so it is easy to use, similar to PPD. It is a point of care. Even it can be used in the remote areas. It does not require any infrastructure. It is specific. And because these two antigens are specific to Mycobacterium tuberculosis, it is not affected by previous BCG vaccination status. So all the countries, TB endemic countries, they give the BCG vaccination at birth for all the newborn babies. So that is again not an hindrance to the testing with the CyTB. So it is an easy replacement for the PPD that is currently in use in most of the countries. Again, HIV infected pediatric population, there is a single cutoff. So in the clinical trials, it has been established that after administration, 48 to 72 hours later, if there is an induration which measures more than 5 millimeter, then it is considered as a positive skin test, and we can say that the individual is latently infected with TB. So this table shows the diagnostic benefits of the CyTB. So during clinical development, you will see this different word, the CTB as well as CyTB. CyTB, so the skin test is approved in India with the brand name of CyTB. And also it is approved in EMA, that is Europe, with the brand name of Siltipsy. So it is highly specific. It is not affected by BCG vaccination, while PPD, it has definitely interpretation with the previous BCG vaccination. Then all these three tests have equal high sensitivity. So single cutoff with the CyTB. As I mentioned, there is a complex test interpretation for the PPD. And IGRA, basically, it requires lab and infrastructure. And moreover, there is one criteria in the IGRA, indeterminate results, where maybe you will have to repeat the test, and that also adds to the cost. While in CyTB, the single cutoff, you can have the diagnosis. Cost-wise, IGRA is costly. Definitely, CyTB would be cheaper as compared to IGRA. The PPD, the availability of PPD is always a challenge. We see the shortage because of complex manufacturing, and there are very less manufacturers. But in terms of CyTB, there is a robust high yield, and we can, the manufacturing is well characterized. We can produce number of doses as required for the world. So in terms of availability, should not be a challenge with the CyTB. And as I mentioned, in the children and HIV positive, it showed the robust results. So this slide shows the clinical development of the CyTB. So the phase 1 clinical trial, it started in 2007. So 2 phase 1 clinical trials were conducted, one in UK, another in Denmark, okay? Then after that, 2 phase 2 clinical trials were conducted, one in TB endemic country in South Africa and another in UK. Basically, during these 2 phase 2 clinical trials, the cutoff of the specificity and the sensitivity was tested, and it was established that the cutoff of 5 mm is diagnostic for the latent TB with the CyTB. And in all these clinical trials, all these 7 clinical trials, the individuals, study participants received CyTB on one arm and the PPD on another arm. So they themselves served as a control with the PPD. And additionally, the blood sample was collected before administration for the QFT testing for individuals 5 years and above. So these trials were conducted from 20 years, babies 20 years old, and no upper limit. So the 5 mm criteria was established with these 2 phase 2 clinical trials. And additionally, it was also established if you give the CyTB now, and if it is negative, can we repeat the CyTB? Yes, after 6 weeks, 4 to 6 weeks after the CyTB administration, we can repeat the CyTB. There is no sensitization with the previous injection. And then 3 large phase 3 clinical trials, 1 in Spain and 2 in the South Africa, they were conducted, and they showed comparability with the QFT, and it was more specific as compared to PPD. In all these 7 clinical trials, more than 3,000 participants were enrolled, and additionally, it included 700 children and around 500 HIV infected. Apart from this, the government of India also conducted clinical trials in 3,500 adults and the children. So there is a large safety data available with this vaccine, with this diagnostic test. So, TB as a diagnostic test, it received the marketing authorization in May 2022, and Government of India is already using TB programmatically in school-going children as well as in adults. Recently, in January 2025, we received EMA approval to use the TB in 29 European countries, which forms part of EMA. And we also consulted WHO, and they said that once you get the approval from the stringent regulatory, we'll have the abbreviated review. So after EMA approval in January, we have submitted our proposal for WHO PQ recently. So we are expecting maybe in a couple of months, we should have the WHO PQ as well for the TB. So after the latent TB, detect latent TB, another part comes is the TB vaccines. So this is again number of TB vaccine candidates. They are developed, some of them after unsuccessful in clinical phases, some of them were withdrawn from the early phases like phase 1, phase 2, some after phase 3 as well. So this slide shows there are around, this is around maybe till August 2024. This slide is based on August 2024. So there are 15 various vaccine candidates in clinical pipeline, out of which 12, these are the 12 vaccines which are in clinical trials, and the 6 vaccines. So the even traditional BCG was evaluated in as a BCG revaccination. For prevention of infection. I guess this trial is also published, and it did not show any effect for prevention of infection as a BCG revaccination. Then GAM TB-vac, it was again prevention of disease. Then in ICMR, that is Government Institute, Indian Council of Medical Research, they conducted prevention of disease, TB disease clinical trials with 2 candidates, one as a IMUVAC, which is actually approved for the prevention of leprosy. Basically, this is a mycobacterium-based whole cell vaccine. And another is a VPM 1002, that is recombinant serums, recombinant BCG vaccine was evaluated by ICMR for prevention of disease. So VPM was evaluated in large 3 phase 3 clinical trials. One is a prevention of infection. That was conducted in, basically that study is ongoing in 5 countries in Africa, and where we are giving RBCG, head-to-head comparison with the traditional BCG vaccination in newborn babies, and we are looking at the prevention of TB infection. Then prevention of disease in household context, as ICMR conducted this trial, and the prevention of TB recurrence. So those TB patients who were newly diagnosed with the pulmonary TB, drug-sensitive pulmonary TB, they completed their 6 months treatment. After completion of treatment, they were vaccinated to see the efficacy of vaccine for prevention of TB recurrence. So these 2 trials for the VPM 1002, POD and POR are completed, and the results are available. So the VPM 1002. So this is a next generation BCG vaccine. It is similar to BCG vaccine. We have just made a couple of changes into the BCG vaccine, and that's why we are calling it recombinant BCG. What are those changes? And so basically, it is a parental strain BCG subtype PROC, same as BCG. So genetic modification. So we have inserted listerolysine gene into bacterial genome of urease C gene. So what it does? So urease C gene is knocked out from the BCG strain. Instead of that, we are inserting listerolysine gene. So what it does? So normally, what happens when the BCG vaccine strain or the wild Mycobacterium tuberculosis bacilli, they go into the phagosome. So they remain into this phagosome and they remain dormant. That's why the TB bacilli or the BCG vaccine can remain in the body for longer duration. And because of that, with the wild infection, there could be a relapse or similarly latent TB infection. So whenever the immune system goes down, it reactivates. But in case of RBCG or VPM 1002, because of this genetic modification, once this vaccine is inside the phagosome, the pH, as urease C gene is removed, the pH goes below 6. It is basically optimum pH of 3 to 5. And it activates the listerolysine gene. And as the listerolysine gene is activated, it breaks the membrane, phagosome membrane. And that's why the antigen, various antigen, vaccine antigen are exposed into the cytoplasm. And that's why as compared to BCG vaccine, which activates only MSC class 1, class 2 pathway, our BCG activates both MSC class 2, MSC 2, as well as MSC 1 pathway. And there is a broad immune response that is expected with the RBCG. That is again an advantage. Moreover, once antigen goes into the cytoplasm, it causes the apoptosis of the cells. And these apoptotic body cell forms, they in turn also help in generating immune response, and the cell dies. That's, so the vaccine induces immune response, and it is cleared from the body. So there are no expected long-term safety issues with the recombinant BCG vaccine. So this slide shows the clinical development of this vaccine, VPM 1002 or the RBCG. So 2 phase 1 clinical trials were conducted in adults. One was done in Germany, another one was conducted in South Africa. And then 2 phase 2 clinical trials were conducted in South Africa in newborn babies in comparison to BCG. So all these trials showed that it is highly immunogenic and safe vaccine. In fact, in phase 2B trial, we enrolled the HIV exposed babies as well. And even in those babies, vaccine was found safe. After these studies, we had a number of phase 3 clinical trials. So the one, as I explained, is the primary clinical trial that was done in Africa for the prevention of TB infection in newborn babies. Then phase 2/3 is a TB post-exposure for prevention of TB recurrence. Another phase 3 is conducted by ICMR in household contacts, that is index TB patients. So the new TB patients in the household, once it is diagnosed, within 1 month, all the family members, close contacts were screened and enrolled into the study. And they were followed for 3 years for TB disease. Apart from this, as you may be aware, the BCG vaccine is used for the treatment of bladder cancer as an instillation. So RBCG was also evaluated in phase 1 and phase 2 clinical trials for this indication, and it has found to be effective. And very soon we will be applying for the licensure for this indication as well for the bladder cancer. Apart from that, during COVID, we also tried these vaccines for prevention of COVID-19. In India, it was conducted in 7,000 participants, as well as one additional phase 3 clinical trial was conducted in Germany. So obviously, we did not find any efficacy against the COVID, but vaccine, we do have a large safety data, and vaccine is safe. So these are the 2 trials I was talking about, the prevention of disease. And this trial is completed, and we found a good efficacy. This was conducted by ICMR. Another one was the prevention of TB recurrence. Here also, we found that the vaccine prevents the symptomatic TB recurrence. And the symptomatic TB recurrence is important. If you can prevent symptomatic TB recurrence, we can again break the spread of the disease. So based on these 2 clinical trials and all previous phase 1, phase 2 clinical trials, we already submitted our dossier to the Indian regulator for the marketing authorization of this vaccine for prevention of pulmonary TB in pediatric population. That is, the trial was conducted in 6 years and above. So we found the efficacy in 6 to 18 years age group. So prevention of pulmonary TB in pediatric population, as well as prevention of extrapulmonary TB in overall general population, 6 years and above. So this diagram shows basically how can we implement this detect, treat, and prevent. So there are healthy individuals, and Some of them, first, if they have any symptoms, suspected TB symptoms, we should evaluate them for the TB, active TB disease. If they are positive, if they have active TB, then give them the treatment, whatever 6 months, based on if they are MDR TB, RR TB, give the complete treatment. Once they complete their treatment, 1 month after that, we give the vaccine. That is recombinant BCG vaccine. If the individual healthy, no TB symptoms, then we screen them for the latent TB infection using CyTB. If they are negative, CyTB negative, then we give them the vaccine so that they don't get the TB disease. If they are CyTB positive, then we give the TB preventive therapy, that is 12 weeks or the 4 weeks as per the national strategy or the national program for each country. Once they complete the treatment, after 1 month after completion of treatment, we give the vaccine, RBCG. Why we are saying the 1 month? Because recombinant BCG vaccine is a live vaccine, and the antibiotics can affect the immune response or might kill the vaccine bacilli. Along with that vaccination, we can give the supplements, vitamin supplements, and they follow them for 1 year for long term. So this can be implemented in the programmatic manner, considering high reservoir of TB maybe in the school-going children initially, and this definitely will help in achieving the targets. So to conclude, WHO as well as all state members of United Nations have committed to end the global TB epidemic. So the 2030 targets were 90% reduction in the number of deaths and 80% in the TB incidence rate. And this can be achieved as with detecting latent TB, treat it and prevent it with the vaccination. This can be important step forward towards the TB world free. This was my last slide. Thank you very much for your attention.